in this video, we are going to discuss mainly about the Ti Plasmid. Okay, and for that there will be two videos. This is going to be the first part, and the next part will be also there. Okay, so let's start with it. So we will start about vector for plants. Okay, now the vector for plants can be a plasmid, it can be a viral genome as well. Now let's talk about the plasmid based vector which we use for plants. Now this Ti plasmid and Ri plasmid, these two plasmids are present in the Agrobacterium species and they are used as vectors for plants. Now this Agrobacterium is a naturally occurring gram-negative soil bacteria, and this Agrobacterium have two common species. The first one is Agrobacterium tumefaciens and the second one is Agrobacterium rhizogenes. Now these two species are known as natural genetic engineers because they have ability to transform plants. Okay, so this for plants, the natural genetic engineers, well, these two are there, the Agrobacterium tumefaciens and Agrobacterium rhizogenes. Now this Ti plasmid, which stands for tumor inducing. This tumor inducing plasmid occurs in Agrobacterium tumefaciens, and this Ri plasmid, which stands for root inducing plasmid, is found in Agrobacterium rhizogenes. Now this wild type Agrobacterium tumefaciens results into crown gall disease especially in dicots. Okay, primarily in dicots, this wild type Agrobacterium tumefaciens results in this disease known as crown gall disease, and it happens when a defined segment of Ti plasmid is integrated into plant genome, and that defined segment that we are talking about is known as tDNA, which is the transferred DNA. Okay, so when the tDNA of this Ti plasmid of this A tumefaciens is transferred and integrated into the plant genome, then that causes this crown gall disease, primarily in dicots. Similarly, this Agrobacterium rhizogenes results into hairy root disease in plants, and the reason for this disease is the Ri plasmid of A rhizogenes, and these Ri plasmid of A rhizogenes is analogous to that of Ti plasmid. Now let's start with Ti plasmid based vector. Now this Ti plasmid is a large sized plasmid whose size is about 200 kb. Now this Ti plasmid have a defined DNA segment which is known as tDNA, and this tDNA is transferred into plant cell. Okay, the tDNA of the Ti plasmid is getting transferred into plant cell, and that is causing your transformation, the resulting disease. Now the size of the tDNA varies, okay, from 15 kb to 30 kb, and it depends on the strain, okay, which strain you are using. If the strain changes, then the size of the tDNA might also change, and that variation is from 15 kb to 13 kb. Now this tDNA is flanked by 25 base pair imperfect direct repeats that are known as border sequences. Okay, so tDNA is flanked by border sequences, and what are these border sequences? Well, their size is 25 base pairs and they are imperfect direct repeats. Now this border sequence which we just saw here, this border sequence is not transferred intact into the plant genome, but it is involved in the transfer process. Now the right border sequence is necessary. Okay, the border sequences are of two types, right border sequence and left border sequence. So out of that, the right border sequence which is there, it is very necessary for the transfer and integration of the tDNA into the plant genome. Okay, so the right border is necessary. If the right border sequence is deleted, then that will result in the abolition of the tDNA transfer. It simply means that if right border sequence is not there, then tDNA transfer will not take place. Now the tDNA that is integrated into plant genome has a precise junction in it that retains one or two base pair of this right repeat. Okay, so there is a precise junction that retains one to two base pair of the right repeat. But if you are talking about left junction, then left junction varies, and it may be 100 base pair short of the left repeat. Now outside the tDNA region, there is this cis element which is designated as overdrive sequence, which is near the right border, and this overdrive sequence or this cis element, okay, it is required for the efficient tDNA transfer. Okay, if you want to do efficiently the transfer of the tDNA into the plant genome, then you should make sure that this cis element or this overdrive sequence is present in your Ti plasmid near the right border. Now, this tDNA carries genes for phytohormones and opines which are expressed in the plant cell. Okay, for the phytohormones like auxin and cytokinin, and for opines there are genes in this tDNA, and those genes are expressed in plant cells. So, in plant cells there is production of opines and auxin and cytokinin, and that production is because of this presence of the tDNA. Now, overproduction of those phytohormones at the site of infection will result into the proliferation of the wound cells, and that will result in the formation of this gall or tumor, and this thing will harbor the population of the bacteria. So that is how it is going to work. The bacteria will infect. At the site of infection, there will be proliferation of the wound cell because there is overproduction of phytohormones, and that whole structure, that tumor will be produced, which is known as gall, and that in that tumor the population of bacteria will then grow. Okay, so this is how the crown gall disease is happening. Now we talked about opines, right? The tDNA have genes for opines, and it is produced in the plant cell. Okay, now these opines are of different types. Nopaline, octopine, and agropine. Okay, these three are the different types of the opines. And the thing is that this Ti plasmid is divided into different groups, and that classification is based on the type of opine. Okay, so which type of opine will the tDNA of the Ti plasmid will be producing in the plant cell? On the basis of that, the Ti plasmid is divided into different groups. Okay, so on the basis of that, the Ti plasmid is of nopaline plasmid, octopine plasmid, and agropine plasmid. Okay, so these are the three classes of Ti plasmid, and this classification is based on which type of opine is there in the Ti plasmid. Now, these opines are the condensation products. Okay, they are when amino acid and keto acid condenses, or when amino acid or sugar condenses, then there is formation of this opines. Okay, so opines are the condensation product of amino acid and keto acid, or they are the condensation product of amino acid and sugars, but not sugar and keto acid. Okay, that is not given. Either amino acid and keto acid or amino acid and sugar. Okay, now this opines serves as the carbon source and nitrogen source, and they are catabolized by agrobacterium. So when in that tumor the agrobacterium is growing, then the carbon source and nitrogen source for the growth of that agrobacterium is the opine, which is produced by the plant cell itself because of the tDNA is integrated in the genome of that plant cell. Okay, so this is how it is going to be. So these are the different genes which are present in your Ti plasmid, and let's discuss about the genes, then their product and their function. So OCS gene is there, which results in the formation of this product, octopine synthase, okay, this enzyme, and this enzyme then results in the opine synthesis. Okay, so OCS, the product is octopine synthase, and this octopine synthase then result in the formation of opine. Okay, and then there is this NOS. Okay, this results in the formation of nopaline synthase, and that causes also opine synthesis. Then there is TMS1, which results in the formation of tryptophan 2 monooxygenase, and this enzyme is responsible for oxygen synthesis. And then there is this TMS2. Okay, and this results in the formation of indole acetamide hydrolase, and this results in the formation of oxygen. Okay, and then there is this TMR, which results in the formation of this enzyme known as isopentyl transferase, and this enzyme results in the formation of cytokinin. And then there is AGS, which results in the formation of this enzyme known as agropine synthase, and which is also responsible for opine synthesis. Okay, so these were the different genes on your Ti plasmid. These were their product, and this were their functions. Okay, so yeah, this is how the Ti plasmid looks like. Okay, there is left border sequence, then there is tDNA, then there is right border sequence, and then in this region is your opine catabolism region, then there is origin of replication ORI, and this big chunk here is the Vir genes, which is also known as the virulence region. So this diagram is representing your Ti plasmid. Okay, so that's all for this video. The discussion will be continued in the part 2. So see you next time. Thank you for watching.